Biological basis of behavior. The brain is where every introductory psychology course begins, and it's also among the first chapters covered by introductory textbooks. We start here because psychologists are interested in understanding behavior, emotions, thought, and consciousness, all of which originate in the brain. In the past 3 decades, the study of the brain's role in behavior, known as behavioral neuroscience or biological psychology, has expanded exponentially, and many new vibrant areas of research have emerged that study behavior anywhere from molecular interactions inside of the individual nerve cells to broad and expansive efforts to understand how large networks of nerve cells give rise to emotions and other experiences of consciousness. As is now widely known, the brain is a massive network composed of billions of brain cells wired together to pass information back and forth. This system is actually a complex triad of architectural, electrical, and chemical components working in concert. In this video, we will describe each component and begin to think about potential projects that will help further develop your understanding of the relationship between the brain and both conscious experiences and behaviors. Working to understand something so incredibly complex necessarily requires some amount of oversimplification at every level. What's important to remember, as parts of the brain are labeled and processes within the brain are defined, is that emergence is always at play. In science, emergence occurs when the whole is greater than the sum of its parts, meaning the whole has properties its parts do not have. A cell, for example, is just a group of chemicals, none of which are alive. But through complex interactions of those lifeless chemicals, life emerges. As is also true of a brain-mind relationship. Not a single one of the 100 billion cells that make up your brain is conscious. And yet, through the complex exchanges of chemical and electrical signals within the vast networks these cells create, consciousness emerges. Architecture. To begin describing the specialized regions of the brain involved in various behaviors, we'll begin with the hindbrain, which controls the most basic functions of life. First, there's the medulla, the region of the brain that adjoins the spinal cord. The medulla controls for breathing, heart rate, blood circulation, and balance. Above the medulla is the pons. The pons controls attentiveness and the timing of sleep and dreaming. Damage to certain parts of the pons can put a person into a semi-permanent sleep-like state. Additionally, REM sleep may also originate in the pons. Inside the pons in the medulla lies portions of the reticular formation, a network of nerves extending from the spinal cord right up through the thalamus. The reticular formation plays a role in autonomic functions of the body, like circulation, respiration, and digestion, but also in pain modulation, sleep, and consciousness. Sitting on top of the pons is the midbrain. This structure helps orient an organism in the environment and guide movement toward or away from stimuli. The midbrain is also believed to help regulate our experience of pain, modulate mood, and shape motivation. Just behind the pons and medulla is the cerebellum. The cerebellum coordinates voluntary movements such as posture, balance, and coordination, resulting in smooth and balanced muscular activity. Damage to this organ can cause problems in spatial reasoning, discriminating sounds, and integrating input received from various sensory systems. More recent evidence suggests that the cerebellum may also help us judge time, modulate our emotions, and even discriminate sounds and textures. Next, the forebrain is made up of brain structures including the thalamus, hypothalamus, amygdala, hippocampus, and cerebral cortex. The thalamus is involved in sleep, wakefulness, and in relaying motor and sensory signals to the cortex. The thalamus also closes pathways of incoming sensations during sleep. The hypothalamus is positioned underneath the thalamus. It has been found to be involved in controlling motivated behaviors such as eating, drinking, and sexual activity, as well as involuntary rhythms such as the sleep-wake cycle and detecting when the body is too cold or too hot. The amygdala is believed to play a role in emotional response, specifically anger, and it is involved in determining whether a stimulus is a threat or not. It helps predict when something frightening is about to happen. The amygdala is particularly active, for instance, when playing video games or seeing fearful faces. Nearby, the hippocampus plays an important role in learning, memory, spatial orientation, and creating new memories. Of all these varied and crucial roles that these many brain regions play, it's the cerebral cortex that receives the most careful consideration and study among behavioral neuroscientists. In fact, the cortex is involved in every thought and perception, as well as our ability to produce and understand language, and to construct and experience emotion. It is crucial in order to believe, organize, remember, and even to hope. Structurally, the cortex is a thin covering on the outer surface of the brain, on average, just 3 millimeters thick. Still, the cortex makes up approximately 80% of the human brain. This enormous volume is made possible by the fact that the cortex consists of a very large sheet of tissue, crumpled up into the limited space inside the skull. This crumpling produces the brain's most obvious visual feature, the wrinkles. Some of the wrinkles are actually deep grooves that divide the brain into different anatomical sections. The deepest groove is the longitudinal fissure, dividing the brain into two halves, the left and the right cerebral hemispheres, which are connected through a thick band of fibers called the corpus callosum. The corpus callosum allows communication between the two hemispheres of the brain. It is responsible for transmitting neural messages between both the right and the left hemispheres. The right hemisphere controls muscles on the left side of the body, and the left hemisphere controls muscles on the right side of the body. This is also true of sensory information. Sensory information from the left side of the body crosses the corpus callosum for processing in the right hemisphere, while information from the right side of the body crosses to the left hemisphere for processing. Therefore, damage to one side of the brain will affect the opposite side of the body. Other fissures divide the cortex in each hemisphere into 4 lobes, rather like continents in the brain. The frontal lobes form the front of the brain, right behind the forehead. The central fissure divides the frontal lobes on each side of the brain from the parietal lobes, the brain's topmost part. The bottom edge of the frontal lobes is marked by the lateral fissure, and below this are the temporal lobes. Finally, at the very back of the brain, directly adjoining the parietal and temporal lobes, are the occipital lobes. Many researchers divide cortical tissue into 3 broad types, located within each of these lobes. Sensory areas receive and interpret information from the eyes, ears, and other sense organs, and the motor areas control our behaviors. Each of the remaining areas are typically called association areas and are said to be involved in many complex processes, including those broadly referred to as thinking. The primary motor cortex is located in the back of the frontal lobe, and stimulating different parts of this area leads to movement of specific body parts. That is, each portion of the primary motor cortex corresponds to a specific part of the human body. This map is often summarized with a motor homunculus, which is a picture showing each body part next to the bit of the motor projection area that controls its movement, with equal-sized areas of the body failing to be controlled by equal amounts of cortical space. Instead, the parts that we can move with the greatest precision, for example, the fingers, the tongue, receive more cortical area than those which we have less control. The shoulder, the abdomen. The primary somatosensory cortex is directly behind the primary motor cortex in the front of the parietal lobe. This area is the initial receiving area for sensory information arriving from the skin senses. As with the motor cortex, space in the somatosensory area is proportional to the sensitivity of the body region, with much more space for lips and fingers, for example, than for larger but less sensitive areas of the body. The brain has similar primary cortical areas for vision and for hearing, and these are located in the occipital and temporal lobes respectively. Notably, the prefrontal cortex has received some of the most intense attention from neuroscientists because of its involvement in planning, decision-making, mood, personality, and self-awareness. Despite ample evidence of seeming specialization within brain regions, it's important to remember that complex mental functions don't reside in any one place in the brain. Complex processes, like memory, language, and attention, result from synchronized activity from many brain areas. The achievements we can easily observe, thinking, feeling, or acting, require exquisite coordination among brain regions. And at the root of this coordination lie the neurons, that is, the communication cells contained within the brain and spinal cord. There are around 10 to 15 times as many neurons in the brain as there are people on Earth, and each of the more than 85 billion neurons receive, process, and transmit messages to thousands of other neurons, with an estimated 160 trillion neural connections in the human brain. The functioning of our brain depends on continual crosstalk within complex networks, or work groups, of neurons. The neuron specializes in sending and receiving information. A description of a neuron typically includes the dendrites, the cell body, and the axon. The dendrites, the input side of the neuron, receive signals from many other neurons. In most neurons, the dendrites are heavily branched. The cell body contains the neuron's nucleus and all the elements needed for normal metabolic activities of these cells. The axon, finally, is the output side of the neuron and sends neural impulses to other neurons. The axon usually extends outward from the cell body, and it may fork into several branches at its end. Neurons come in many shapes and sizes. Their cell bodies vary from 5 to about 100 microns in diameter. In comparison, the average human hair has a diameter of about 100 microns. Neurons' dendrites are typically pretty short, say a few hundred microns. Axons, however, can be much longer. For example, the longest axons in humans are those of the motor neurons, which transmit neural impulses from the brain to the muscles. As a result, these particular neurons, with their small cell bodies connected to very long axons, have roughly the same proportions as a basketball attached to an electrical cord 1.5 miles long. The brain also contains another type of cell, called glia, whose function is just beginning to be understood. For many years, scholars thought glia only played a few roles, holding the neurons in place and supplying them with nutrients and oxygen. Turns out, however, that glia cells have a rather broad set of vital functions. First, Glia do provide nourishment for the neurons, but also play a role in controlling the nutrient supply. For example, the sugar glucose is the main fuel for the nervous system, but most of the energy that neurons need does not come from glucose directly. Instead, the glia convert glucose into another molecule called lactate that feeds the neurons. The glia cells are also sensitive to the activity level in each neuron and increase the blood flow, providing more oxygen and fuel whenever the neurons in the brain region become more active. Additionally, glia cells are involved in the brain's development. Before birth and in the months afterwards, the human brain grows at a remarkable rate. As its cells rapidly reproduce and differentiate, the newly created neurons then migrate from one position in the brain to another, moving at a speed of up to 1 millimeter each day. This migration is guided by glia cells, acting as guide wires. Then, once the neurons have reached their destinations and established the appropriate connections, the glia produce chemicals that help shut down the process of neural growth. In this way, the glia ensures a relatively stable pattern of connections. The glia may also have other jobs to do. For example, several recent studies suggest that glia can talk back to neurons, sending signals that help regulate the strength of connections between adjacent neurons. In some circumstances, glia can also release chemicals that increase the reactivity of neurons. The neuron is in some ways just a cell. It has a nucleus and a cell membrane that defines its boundary. What makes a neuron distinctive, though, is its sensitivity to stimulation. When stimulated, a cascade of changes are produced that result in an electrical signal called an action potential. This signal, sent from one end of the neuron to the other, is the neuron's main response to input, as well as the fundamental information carrier of the nervous system. When a neuron is at rest, there is an uneven distribution of ions across the cell membrane. When a neuron is sufficiently stimulated, an action potential is generated and travels along the axon to the terminal branches by means of a change in polarity across the membrane of the axon. In response to a signal from another neuron, positively charged sodium and potassium gated ion channels open and close. The positively charged sodium ion channels open at the beginning of the action potential, and positively charged sodium ions move into the axon, causing depolarization. Repolarization occurs when the positively charged potassium channels open and the positively charged potassium ions move out of the axon, creating a change in polarity between the outside of the cell and the inside. In other words, during an action potential, positively charged ions flow rapidly into the neuron, and then, just as rapidly, flow out. This electrical impulse travels down the axon in one direction only, to the axon terminal, where it signals other neurons. When an action potential occurs, we can describe it as the neuron firing. Although the flow of ions in or out of the neuron is fast, even so, the full movement of the action potential across the neuron is surprisingly slow, in that it travels at about 1 meter per second. If this were the top speed for neural signals, it would be a serious problem for most organisms, because most fast-paced actions would be impossible. Fortunately, yet another function of glial cells is to increase the speed of neuronal communication. The glia that accomplish this are mostly made of a fatty substance known as myelin. Soon after birth, these glia cells start to wrap themselves around the axons of neurons, especially the longer axons that span greater distances and require greater transmission speed. This myelin sheath covers a portion of the axon, and soon the entire length of the axon is covered by a series of these wrappers. Crucially though, there are gaps called the nodes of Ranvier between the successive wrappers, and it's this combination of wrappers and gaps that speeds up the nerve impulses traveling along these myelinated axons. If an axon is myelinated, ions can move into or out of the axon only at the nodes of Ranvier. At all other locations, the axon is enclosed within its myelin. and this blocks ion flow. Therefore, in essence, the action potential has to skip from node to node, and thanks to these jumps, it moves more quickly at speeds up to 120 meters per second. It is also important to mention that the neuron either fires or it doesn't. There's no in between. Though we need some way to differentiate the weak signals from the strong. This is partially accomplished by more intense stimuli exciting a greater number of neurons. This happens because neurons vary tremendously in their excitation thresholds. As a result, a weak stimulus stimulates only neurons with relatively low thresholds, while a strong stimulus stimulates all of those neurons plus others whose threshold is higher. Chemical messages. When the action potential reaches the axon terminal, it triggers the release of chemicals known as neurotransmitters into the synaptic cleft, which is a fluid-filled gap between neurons that's less than 1/millionth of an inch wide. Neurotransmitters from this presynaptic neuron are taken up, that is, bind to specific receptor sites on dendrites of nearby neurons, stimulating them and starting their action potential. So, what happens to the neurotransmitters after they've affected the postsynaptic neuron? They can't just stay where they are because they might continue exerting their effects long after the presynaptic neuron has stopped firing. To avoid this problem, some neurotransmitters are deactivated by special cleanup enzymes that break them up into their chemical components. More commonly, though, neurotransmitters are not destroyed, but they're reused. In this process, called synaptic reuptake, the neurotransmitter molecules, after they've had their effect on the postsynaptic cell, are rejected from the receptors, vacuumed up by the molecular pumps back into the presynaptic axon terminals, and repackaged for future use. Through this process, one individual neuron influences thousands of others, and thousands of neurons can influence one, all simultaneously. This is the brain in action. In any given moment, our brain is working to generate just the right balance of neurotransmitters. At night, for example, the brain needs to increase rest and recuperation neurotransmitters level to transmit certain thoughts in a calming, tranquil, and relaxing way to induce sleep. Examples of these neurotransmitters are melatonin and serotonin. In the morning, the brain must lower its rest and recuperation neurotransmitter levels and increase its excitatory neurotransmitter levels for us to function consciously. Examples of excitatory neurotransmitters are acetylcholine, glutamate, aspartate, noradrenaline, and histamine. In stressful situations, it must increase levels of fight or flight transmitters, for example, epinephrine and norepinephrine, that help you to remain calm and in control. It's critical that all of the major neurotransmitters are present in sufficient amounts all the time. When there are insufficient amounts of neurotransmitters, it upsets the chemical balance of our brain. Hence, some symptoms may be experienced, including depression, lethargy, hopelessness, mood swinging, anxiety, panic, irritability, insomnia, and aggression. Importantly, neurotransmitters can be classified by function. Excitatory neurotransmitters. These types of neurotransmitters have excitatory effects on the neuron. They increase the likelihood that the neuron will fire an action potential. Some of the major excitatory neurotransmitters include epinephrine and norepinephrine. Inhibitory neurotransmitters. These types of cells have an inhibitory effect on the neuron. They decrease the likelihood that the neuron will fire an action potential. Some of the major inhibitory neurotransmitters include serotonin and GABA. Now, some neurotransmitters, such as acetylcholine and dopamine, can have both excitatory and inhibitory effects, depending on the type of receptors that are present. Glutamate and GABA are the most common neurotransmitters in the central nervous system. Neurons in virtually every brain area use these two chemical messengers to communicate with each other. Glutamate rapidly excites neurons, increasing the odds that they will talk with other neurons. The release of glutamate is associated with enhanced learning and memory. When abnormally elevated, glutamate may contribute to schizophrenia and other mental disorders because in high doses it can be toxic, damaging neural receptors by overstimulating them. GABA, in contrast, inhibits neurons by dampening neural activity. That's why most anti-anxiety medications bind to GABA receptors. They tend to suppress overactive brain areas linked to worry. GABA is considered an absolute workhorse in our central nervous system, playing critical roles in learning, memory, and sleep. Beyond an excitatory or inhibitory effect, there are also various ways to enhance or impede the actions of a neurotransmitter by introducing chemicals in the form of medications. Chemicals that enhance a transmitter's activity are called agonists. Those that diminish a transmitter's activity are called antagonists. Agonists exert their influence in many ways. Some agonists actually mimic the neurotransmitter, so on their own, they can activate the receptors. Other agonists block the reuptake of the transmitter into the presynaptic cell. And still, others work by counteracting the cleanup enzyme that breaks down the transmitter after it has triggered a response. Both of these mechanisms have the effect of leaving more transmitters within the synaptic gap. This increases the transmitter's opportunity to influence the postsynaptic membrane, and so ends up increasing both the strength and the duration of the transmitter's effect. Antagonists work through similar mechanisms, but with the opposite effect. Thus, some antagonists prevent the transmitter from working by binding themselves to the synaptic receptor and blocking off the transmitter, essentially serving as a kind of putty in the synaptic lock. Other antagonists operate by speeding up reuptake, and others by augmenting cleanup enzymes. Cocaine, for example, is an agonist. It works by blocking the reuptake of dopamine, norepinephrine, and epinephrine into the presynaptic molecules. The effect is arousal throughout the body, restlessness, and in some cases, euphoria. Many antidepressant medications, including Prozac, Zoloft, and Paxil, work in roughly the same way, but specifically block the reuptake of serotonin. Still, other drugs are antagonists. Some of the medications used for schizophrenia, for example, block postsynaptic receptors and seem effective in helping patients control psychotic thinking and restore normal functioning in their lives. Opiates, such as codeine and morphine, also function as agonists, increasing receptor site activity. They reduce our emotional response to painful stimuli by binding with opioid receptors and mimicking endorphins. Tranquilizers such as Xanax, whose generic name is alprazolam, diminish anxiety by stimulating GABA receptor sites, thereby driving down neural activity. Another key idea is that individual neurons are selective in what neurotransmitter they will respond to. Many neurons are responsive to more than one neurotransmitter. But even so, Each neuron has its own pattern of sensitivities. For example, a neuron inhibited by GABA will respond differently, or perhaps not at all, to molecules of serotonin that happen to float by. This notion of receptors being selective in their response and the idea of different neurotransmitters providing different signals are key elements in controlling the complex flow of information throughout the brain. Though the neurotransmitter system has provided some evidence specificity at the molecular level, it's important to remember that a mental event, thought, perception, memory, or emotion is not created by only one set of neurons or one type of neurotransmitter. Instead, combinations of different neurons and associated neurotransmitters can create similar instances of these experiences. Neuroscientists call this principle degeneracy. Degeneracy means many to one. Many combinations of neurons can produce the same outcome. In the quest to map the brain, degeneracy is an important, humbling reality check. To be humbled even further, the opposite is also true about the brain. Along with degeneracy, many parts of the brain serve more than one purpose. The brain contains core systems, or networks of neurons that participate in creating a wide variety of mental states. A single core system or network can play a role in thinking, remembering, decision-making, seeing, hearing, and experiencing. A core system is one to many. That is, a single brain area or network contributes to many different mental states and behaviors. So, from this brief description of the structure and function of the brain, it is clear that neuroscientists have made enormous progress, but there are also many, many miles to go. Within the field of psychology, we have the opportunity to learn about many other perspectives and types of analysis that help us to understand our behaviors and mental states. Schwartz and colleagues, in their recent article in American Psychologist, actually warn against what they call neuroseduction, a tendency to accept dubious claims when couched in terms of neuroscience. In one study, undergraduates judged logically flawed reports of research findings to be more convincing when the phrase "brain scans show" was used. Likewise, the company Lumosity seduced more than 70 million subscribers with its brain training games that were supposedly based on neuroscience. That is, until the Federal Trade Commission found the company guilty of false advertising and slapped it with a hefty fine. Even if claims about the brain and its role in behavior have evidence to back them, it's not unusual that they represent an as yet still necessary oversimplification of the vast complexity that our brains actually hold.